working on that chapter 5 homework. There's lots of study helps, especially for chapter 5 and into chapter 7, our genetics. Lots of animations, lots of practice questions. So take advantage of that in mastering. Okay. All right, so to finish up replication. So last time we talked kind of big picture, as well as we talked about lots of enzymes and their role in copying the DNA of a cell. RNA polymerase 3, primase, DNA polymerase 3, DNA polymerase 1, primase, ligase, helicase. You gotta know what all those enzymes do, what their job is. One more that we need to mention that's involved, it's very important, involved in DNA replication is DNA gyrase. Okay, so remember, helicase is the enzyme that we say unzips the DNA, so cuts those hydrogen bonds between the complementary base pairs of DNA and kind of opens up the replication fork. That's helicase. What gyrase does is relax supercoils here upstream of that replication fork. So what are supercoils? I think this is a pretty good illustration of that. There it goes. So if you're winding up two pieces of twine or rubber band, say for example, and you try to kind of open up, separate the two strands, okay, they just get more and more tightly compressed over here, and they kind of bunch up and tangle up. Same principle if you're spinning on a swing. At some point, right, it's getting twisted up so tight, you're bending down more and more, and you can't pull those two chains apart anymore. Okay, that happens to DNA, basically, when you open up this replication fork, and eventually, you can't open it up anymore because of the mechanical. So this enzyme, DNA gyrase, it's necessary to relieve those supercoils so that the replication fork can continue and the DNA can be copied. That enzyme does a lot. Some enzymes, make one little cut and that's all they do. It actually cuts the DNA, both strands. It unwinds the helix just a turn, and then it seals those strands back up. And it's a lot more complicated than that, but that's basically what it does. We have enzymes that do that in our bodies as well, but they are different enough that we actually have antibiotics that target DNA gyrase. The fluoroquinolines and the quinolines, a group of antibiotics that we'll talk about in a few weeks, they basically disabled DNA gyrase. So we will. Sorry, say that again?
Is it specific to microbial replication?
So is it specific to microbial replication? So yes, this enzyme is bacterial. Yep. And just a few other things to mention about replication. Something that happens eventually is that the DNA becomes methylated. So that means methyl groups are added, these CH3 groups. And in bacteria, the methyl groups are typically added to the adenine bases. This happens in humans as well, although cytosines are methylated instead of adenine. So the upshot is that bacterial DNA actually is a little bit different because of the methylation of different bases than what we see, say, in eukaryotic DNA. The purpose of that methylation, there's several different purposes. For one thing, we think it defines the origin. Remember, the origin is the place in the chromosome that replication begins. So we think that maybe is a highly methylated spot there, and that's how the cell knows where to begin replication. It also is involved in DNA repair, and we're going to touch on that probably next time. But cells have lots of mechanisms to make sure that the DNA gets copied correctly, because right? It's the blueprint. If the blueprint is wrong, then all the proteins that are ever made from that DNA will be wrong. So you got to make sure the blueprint is right. If DNA polymerase 3 accidentally puts the wrong base in, so it's mismatched, say you have a G with a matched up with a T, for instance, we have enzymes that come through and fix that. How do they know which base to fix? Basically, one strand is going to be more methylated than the other. The old strand is highly methylated. It takes a little bit for the cell to methylate that new DNA. So the enzymes, again, it's a way for them to know which strand to fix. Okay, and we think there might be some function in regulation. DNA that has methylated its certain parts might be more likely to get transcribed. Okay, so just kind of a general thing there. All right, so thinking big picture for a minute before we move into transcription. All right, so the flow of information in a cell, we call this the central dogma of genetics. Okay, an organism's genotype, that's basically just the sequence of bases in the DNA, okay, in the double-stranded DNA. And of course, when that DNA is copied, we call that replication. Replication doesn't necessarily have to take place before the DNA is transcribed. The DNA gets replicated whenever the cell divides. But then it can be transcribed over and over and over again. It doesn't have to be brand new DNA to get transcribed. Okay, but when the DNA is used as a directions to make RNA, we call that transcription. We're going to talk about the different kinds of RNA here in a minute. But again, following the flow of information in a cell that gets transcribed into messenger RNA. And every 3 of these base pairs, we have a special name for that. We call this a codon. Okay, so the codons are on the messenger RNA. And that messenger RNA then gets read at the ribosomes and translated to a new language, a language of amino acids, which form proteins, polypeptides. Okay, and that is the phenotype of an organism, basically what proteins it has. Okay, so when you think about the phenotype as observable traits, like for us, it's the color of your eyes or how tall you are or whatever. For bacteria, the phenotype might be, can it utilize lactose or can it not utilize lactose? Does it have the enzymes to use lactose as a nutrition source? Okay, so genotype dictates phenotype. What do I say? All right, so let's look at the structure of RNA. This should be review, so we'll go through this fairly quickly. Of course, RNA is single-stranded, where DNA is double-stranded. DNA only has a primary sequence, so we're just looking at a primary structure, right? A sequence of DNA, of nucleotides and bases. RNA can also have secondary and tertiary structures. So RNA can have some more complex folding into beta and alpha helices, for example, for secondary structures. And tertiary structures just means, is that like just people talking, or is that a microphone? Or I wonder if, can you close the door? That might help. Thank you. Okay. All right, so tertiary structures are just more complex structures, and we'll see that not in messenger RNA, but some other kinds of RNA. Thank you. All right, and then of course the nucleotide bases. We don't have thymine in DNA. We have uracil instead. The uracil binds its complementary base pair is adenine, and guanine and cytosine, they are complementary. So 4 bases. And then of course we have the ribose sugar instead of deoxyribose. And so here's the sugar portion, whoops, here. That's the ribose. The only difference between ribose and deoxyribose is right here. DNA does not have this oxygen here. It just has a hydrogen. Okay, and ribose has a hydroxyl group there instead of a hydrogen. So really close to being the same structure as DNA there. Again, the sugar is just really, really close to being the same thing, pentose sugar. Okay. All right, so different types of RNA. We have talked about RNA primers already. Like this. Okay, remember they are between 10 and 20 base pairs long, and they are necessary for replication to begin. That DNA polymerase 3 needs that a 3' hydroxyl group, so a primer, RNA primer gets added there. It's just temporary. So in terms of the amounts of RNA in a cell, primers are a very negligible amount of the total RNA. Again, because they're very little, very small, short, and they don't last very long. Messenger RNA is only about 4% of the total RNA of a cell, which is kind of surprising because to me, that's kind of like the most famous kind of RNA, but it's fairly transient. If it hung around for a long time, then it would just keep getting translated and translated. And if the cell needs to kind of dial in how much protein it needs, then apparently, you know, that's a good way to do it. Just have some messenger RNA, again, that degrades fairly quickly. Okay. Some terms that we need to get used to. Messenger RNA is polycistronic in prokaryotes and monocistronic in eukaryotes. Okay, so that term cistron means gene. Okay, so many genes. Yeah, question? Oh, sorry, necessary for replication to begin. Yeah, thank you. Okay, so many genes in prokaryotes. That just means that messenger RNA can encode for multiple proteins. That doesn't happen in eukaryotes. Their messenger RNA only encodes for one protein. And there may be a few exceptions there, but this is generally true for the most part. Monocistronic, one gene. So one piece of messenger RNA encodes for one protein. Okay, so this is an efficient way for prokaryotes to regulate the amounts of proteins that get eventually produced. So if you have a pathway, you need 3 different enzymes to get the product. It makes sense to get the same amounts of those 3 enzymes. Okay, so we'll get to operons eventually here. Okay. tRNA. About 10% of the RNA in the cell is tRNA. It's pretty short pieces of tRNA, it's about 75 base pairs long, okay, or ribonucleotides in length. Okay. The tRNA's job is to deliver amino acids to the ribosome. Okay. And which amino acid they bring, of course, is based on the messenger RNA sequence. Okay, so we'll look more closely at that when we look at the process of translation. Okay, but we'll just take a minute and look at the structure here of tRNA. Of course, the way you've usually seen it drawn out is part A here. These 3 cloverleafs or a cloverleaf pattern, or this is typically called a hairpin loop. 3 hairpin loops. And these 3 bases down here at the bottom, hairpin loop, we have a special name for that. That is called the anticodon. Okay, so that's going to match up with the codons of messenger RNA, and the amino acid is going to be attached up here at this end of the tRNA, and it starts building the proteins at the ribosomes. Okay, your book always, so this is the way we typically draw the tRNA, but your book recognizes that that's actually not the way it's folded up in nature. It actually, we think it looks something more like this. So whenever your book draws tRNA, they use this icon, this kind of bent over icon to denote some transfer RNA. Okay. All right, and then the last type of RNA, the kind that people tend to forget about, is ribosomal RNA. But this is actually the dominant RNA molecule in a cell. About 85% of a cell's RNA is ribosomal RNA. Of course, where do we find it? It makes up the ribosomes. And an interesting thing about ribosomal RNA is that it can be catalytic. In other words, it can act as an enzyme. Okay, so within the ribosome, that ribosomal RNA catalyzes lots of reactions within protein synthesis, like the amino acids, when they bond to each other, that's catalyzed by ribosomal RNA. And when it does act as an enzyme, we call it ribozyme. Okay, so ribozyme, again, catalyzes many aspects of protein synthesis. Okay. All right, so just a reminder, structure of ribosomes. So remember, prokaryotic ribosomes are considered 70S, eukaryotes 80S. Looking at here, like there's the large subunit, the small subunit. This 16S ribosomal RNA is the dominant molecule in the small subunit of the ribosome. It's associated with lots of proteins as well. But it's interesting how we draw this here. We draw this as you know, a little line like this. Okay, this is what we actually think it looks like. Okay, and I didn't post this. I just wanted you to appreciate the complexity here. But again, this is the 16S ribosomal piece that makes up the small subunit in the bacteria Thermus aquaticus, something like that. So again, it's not just a little line, it's very complex. Here, the tan portion, that's the 16S ribosomal RNA associated with the purple proteins. So again, just that's what it actually looks like, even though that's not often the way we picture it. All right, so let's concentrate on transcription and the events of making RNA from DNA. Okay, we typically describe transcription in 3 steps: initiation, elongation, and termination. Okay, it's just a little bit more complex transcription than replication, because in bacterial cells, basically all the DNA gets copied. It's not really very complicated where it starts and where it stops, because everything gets copied. In transcription, only part of the DNA is getting made into RNA. So basically the initiation, there's all sorts of cues and things that have to happen for the RNA polymerase to bind to a certain region on the DNA for it to start transcribing, to get the right signals to do that. Elongation is very straightforward. That basically we've got the triphosphate ribonucleotides this time. They're carrying with it the energy to polymerize into messenger RNA. And then termination, there's a couple of different ways that happens too. So it's very easy to then get transcription mixed up with translation, because we describe translation in 3 steps like this too. So just try to keep the steps kind of straight in your mind as much as you can, and that will probably just take some repetition. To me, it's not terribly satisfying how your book portrays transcription. So I brought in a couple other graphics that I think helps. Sometimes the book gives so little detail that it doesn't really make sense. So hopefully with a couple of extra pictures, it won't confuse you, but hopefully make it make a little bit more sense. So first of all, keep in mind that in transcription, only one strand or the other of DNA is used as a template, not both of them. So you can't tell from the DNA by looking at it which strand is used. The cell knows, the enzyme knows, and say this strand down here on the bottom, say that was the one that was used to be transcribed. Well, we have names for that. We can call it the antisense strand, or sometimes we call the negative sense strand. And that makes sense because what's transcribed here, what's complementary to that strand is going to be called the positive sense, okay? Messenger RNA that's positive sense is in a form that's able to be read at the ribosomes. And that will make a little bit more sense when we talk about viruses, because there are viruses that carry with them negative sense messenger RNA, negative sense single-stranded RNA. That is not RNA that would be read at our ribosomes. They have to change that into positive sense. Okay, so if, again, if this is, whenever you copy a strand, you get the opposite. If it's positive and you copy it, you get negative. So if this is the negative sense, so this would be the positive sense strand of the DNA. The other names that we have, the other name we have for the antisense strand of DNA, which hopefully makes sense, we also call this the template strand. It's used as a template for the messenger RNA to build off of, okay? This strand up here that's not used as a template then is called the coding strand. And that's a little bit more confusing in my mind to remember. Let's write in some pretend sequences here. So and that will help understand that. So if we've got bases CC, AGT, yes. So the negative strand is the template, and the positive strand is the coding. So let me illustrate why we call it coding, because those two things kind of sound like the same thing. But if this was a sequence of the DNA here, just pretend that it was, then the other strand of DNA would be complementary to that. We'd have GG, TCA. Okay, and then if you look at what the messenger RNA, the new sequence would be here. This would be GGU instead of TCA. All right, so what do you notice about the messenger RNA compared to the coding strand of DNA? It's the same thing, except we have uracil instead of thymine. Okay, so that's why we call it the coding strand up here, because it's going to look just like the new messenger RNA. Oh. So I know we have lots of names for things, then it just makes things a little bit more confusing sometimes, but it's important to kind of recognize. All right, so if you look at the blow up down here, then our main enzyme that's doing the work here is RNA polymerase, and that has first bound down here at the promoter. And look more carefully at how it does that. And then it's adding the triphosphate ribonucleotides on again at the 3' end. So it's growing, the new strand is always adding on to the 3' end. Okay. All right, and I tried to write this down here. That's the template strand down there. Okay. So let's just take a second and look at that main enzyme of transcription, the RNA polymerase. And in prokaryotes, there's just one enzyme that makes all kinds of RNA. So we could be transcribing tRNA or ribosomal RNA. For eukaryotes, they have a specific enzyme. They've got one enzyme for messenger RNA, a different enzyme for tRNA, a different enzyme for ribosomal RNA. But let's compare that RNA polymerase, again, with the replication enzyme DNA polymerase 3. Okay, of course, DNA polymerase 3, it needed helicase in order to unzip those two strands or separate those two strands of DNA. RNA polymerase actually does that itself. Unzips DNA itself. So it has a helicase function right built in with it. Of course, for the DNA polymerase 3 needed an RNA primer to get started. RNA polymerase, no primer necessary. Okay. Of course, DNA polymerase 3 uses deoxyribonucleotides. And RNA polymerase ribonucleotides. Of course, it uses uracil instead of thymine. I also just mentioned, I guess I didn't leave myself enough room really to write this in. DNA polymerase 3, we talked about, has really good proofreading function. It makes an error about 1 in 10 billion times, I think was the number that I used. So that's good. Again, we need the blueprint to be correct. RNA polymerase is not that good. It proofreads a little bit, but it's a lot less efficient. And it makes an error about 1 in 10,000 times. So I won't write that down. You don't have to have that number in your head. Just kind of remember it makes a few more mistakes. But again, that's probably okay. Probably the next time that DNA was transcribed, it would be done correctly. So usually that's not the end of the world. All right, so let's look a little bit more closely at the initiation of transcription. So again, this is a figure that's not in your book, but at least to me, it was helpful to really understand what was going on. All right, so orient yourself here. This line here is supposed to signify the DNA. Okay, and this region, this promoter region, is what we say upstream of the start site. So basically, the DNA that actually gets transcribed into RNA is going to be up here. Okay, but the binding site for the RNA polymerase is called the promoter region of the DNA. And there's some spots on the DNA, some sequences of DNA that are especially important for the RNA to recognize in order to bind tightly. Let me just write that in. So the more tightly RNA polymerase binds to the promoter, the more likely the DNA gets transcribed. Okay, if the RNA polymerase only binds on there fairly loosely, then it's not as likely that that piece of DNA is going to get transcribed. So that's kind of one way that the cell can regulate how often that piece of DNA gets transcribed. One thing that helps the RNA polymerase bind is this sigma factor. Okay, so sigma factor is a protein that helps RNA polymerase bind to the promoter. So Greek letters actually have capital and lowercase. I didn't realize that until not very long ago. So this is a lowercase sigma, all right? We call that sigma factor. This is a prokaryotic thing. Again, it binds to that RNA polymerase and helps it, again, bind tightly to that promoter region. So cells have different sigma factors depending on maybe how often they want that gene to be transcribed. The third thing that you want to look at here is there's two particular regions, the -10 region and the -35 base pair region. We think that those two sequences are particularly important for binding of RNA polymerase. And this -10 region is so important that we have a special name for this sequence. It's called the Pribnow box. Okay, and I'm not sure if your book uses that term. I might mention it once. And that's, again, in prokaryotes only. We use that name. In eukaryotes, they call this the Tata box. So you may have heard of that. Again, these are two regions. We'll say the -10 and -35, important for RNA polymerase binding. In the next slide that I want to show you is a little bit overwhelming. There's a couple things, a couple points that I want to make from this. This is basically, it's not labeled very well here, the sequence of promoters of the operons of E. coli. Okay. What I like about this picture, even though it looks terribly complex, is that it reminds us what we're dealing with here is a sequence of bases in the DNA. So often draw DNA as a line and we kind of forget what that actually is. So each one of these is a base and a nucleotide of the DNA. And I want you to concentrate here just on the top row. This is the sequence of the lac operon promoter. Okay, so that's probably the one you've heard of. Remember, an operon, what that is, it's a group of genes that are regulated together. And this is a prokaryotic thing because prokaryotes are polycistronic. Okay, so it turns out E. coli has like 200 some operons that have been described. Again, the lac operon is probably one that you're more familiar with. We're going to talk about that one later, as well as this trip operon. But again, if you look at the sequence of the nucleotides in the promoter region, the ones that we said were important, that -10 region, or the Pribnow box, that's highlighted in orange. And here's the -35 region. I guess it doesn't get a special name, but that's highlighted in blue. The point here is we think that the sequence of those two regions matters. If you look at those sequence of bases for all of the operons, they've come up with a consensus sequence where, well, most of the time that first base is a T, and most of the time the second base is a T. And we think the more closely those operons, see that -35 region, the more closely it resembles the consensus sequence, the more likely the RNA polymerase is to bind. Okay, so if it matches that consensus sequence, again, then RNA polymerase more likely to bind. Okay, so that's just the point I'm emphasizing, that those bases, it matters. And again, the more likely RNA polymerase is to bind, then the more likely that transcription will actually take place. Okay. All right, so once that RNA polymerase binds tightly and it's free to move down the transcript, then the next step in transcription is elongation. So this is a figure from your book. This is a little bit more straightforward. Basically, this is the sigma factor. It's falling off now, the RNA polymerase. Its job is done. And it's just adding those triphosphate nucleotides. Remember the ATP, those 3 phosphates get cleaved off to make this a really fast polymerization reaction. And again, it's adding on to that 3' end. Okay, and it just moves down, again, moves down the DNA strand. And this figure just reminds us that multiple RNAs can be transcribed all at once from the same tRNA. So you can have multiple RNA polymerase enzymes binding the promoter kind of one after the other and getting transcription of lots of messenger RNA. And this happens in both eukaryotes and prokaryotes. This is happening in eukaryotes in the nucleus, because that's where the DNA is. In prokaryotes, this is going to be out in cytoplasm, because the DNA, again, is not enclosed in a membrane for prokaryotes. All right, how does it stop? There's 2 methods of termination for prokaryotes, and we think that these 2 methods happen in about equal chances of either one of them happening. First one depends on this protein called Rho. So it's Rho dependent, it depends on this Rho protein, which actually is a helicase enzyme. And remember what helicases do, they break those hydrogen bondings between complementary base pairs. So basically this protein kind of moves down the new RNA strand and basically breaks all these kind of temporary hydrogen bonding that takes place when the new RNA is synthesized, and makes the RNA polymerase dissociate from the DNA. Here we've got our new strand of RNA, and that's the Rho, okay? So basically, I've read a lot more about this because, you know, to me, this isn't very satisfying how this really works. And what I've discerned is it gets really complicated, and there's a lot we really actually don't understand about this. So if you want to get in more detail here, you're welcome to try to look that up. But like I said, it gets really complicated. So the basic crux of this involves this Rho protein. The other way that we think termination ends is Rho independent, meaning it doesn't have anything to do with the Rho protein. So we have other names for this. It's also called sequence dependent, or sometimes it's called self-termination. Okay, but those are all referring to the same thing, that it doesn't have anything to do with that Rho protein. So with this method, here's the DNA. And so the RNA polymerase is up here somewhere. Basically, there's sequences in the DNA that are inverted repeats of each other. And I'll explain that in the next graphic, explains it a little bit better. But because there's inverted repeats in that sequence, when the messenger RNA is transcribed, sometimes they are going to kind of bend back on each other and match up here and form a hairpin loop. Okay, that hairpin loop imposes some mechanical stress, or in here it says tension, on the transcript and the RNA polymerase. And that, together with this upstream, or this sequence here of uracils and adenines, remember the U's and the A's only have 2 hydrogen bonds, the G's and the C's have 3, so this is a little bit more loosely bonded. So coupled with those loose bonds and the mechanical stress here caused by the hairpin, it causes the RNA polymerase to dissociate from the DNA and the new transcript. Okay, so this next graphic, which is not one from your book, if you're a little bit more interested here or want to picture what's going on, this does a little bit better job of explaining the inverted repeats. And again, it just shows a little bit better how those can match up with each other and kind of cause the whole new transcript to dissociate from the DNA. Okay. All right, so if we compare differences, step back for just a minute and compare differences in prokaryote and eukaryote transcription. Let's see. Okay, where does it happen? I just mentioned this a minute ago. Of course, in prokaryotes, this is happening in the cytoplasm. In eukaryotes, where the DNA is in the nucleus. The complexity of both of those processes is just way more complicated in eukaryotes. Like I mentioned, eukaryotes have a different enzyme for every kind of messenger RNA there is. And in eukaryotes, there are things like transcription factors and elongation factors. Lots of proteins that are necessary for the initiation and even the elongation of the RNA transcript. Okay. They don't have sigma factors, but they have these other factors in eukaryotes. And processing, I'm not sure if I gave myself enough room here, but I'll try to write this a little bit smaller. In eukaryotes, there has to be some extra processing after the messenger RNA is terminated. There is a 5' guanine cap added. So these are basically altered guanine nucleosides that are added onto the 5' end of the new messenger RNA, or sorry, RNA. And the eukaryotes also add on the 3' end some polyadenylation. Okay, so a stretch of ribosomal RNA that has only adenine bases. And the reason that this is necessary is because the messenger well, whatever RNA, usually if we're talking messenger RNA, that transcript has to travel out in the cytoplasm to get to the ribosomes for eukaryotes. Okay, for prokaryotes, it's already right there in the cytoplasm, but the eukaryotes, it has to travel. So that extra little bit added on each end of the transcript basically protects the transcript from endonucleases that would degrade it. Okay, so again, has to be a little bit more protection there for the eukaryotes. And lastly, For the eukaryotes, not all of that messenger RNA actually gets translated. There has to be some splicing that takes place. The eukaryotes, the introns, also called intervening sequences. I don't know if I spelled that right. Are gotten rid of, or we'll say cut. And the exons, expressed genes, okay, are going to remain. Okay, so think of some way to keep that separate, introns and exons. Which one, again, if you remember intervening sequences, that's the ones that the cell doesn't need anymore. Those get cut out, and the exons actually get expressed, okay? And here is a nice picture that illustrates some of that from your book. Okay, so again, this is in eukaryotes. They get kind of a, what's called a pre-messenger RNA after transcription, where the exons and the introns are still there. Here's the 5' cap, the poly A tail that's added. After some processing by a group of enzymes called spliceosomes, those introns are cut out, and now you have the messenger RNA that's ready to travel to the ribosomes and get translated into proteins. And here's just one other figure that I pulled up from another source, because I like this, because this reminds us that that splicing doesn't always happen appropriately. Sometimes a cell can use different splicing enzymes and make slightly different messenger RNAs end up from that, and sometimes that's a form of regulation. But sometimes a cell makes errors in splicing. So you can see here, if the introns part here, if just all those lines are cut out, we come up with this messenger RNA. If, say, one of these exons was actually cut out, maybe by accident, we would get a little different strand. Over here, if we got maybe part of an exon cut out, the messenger RNA would look like that, which would give us 3 different protein products. And again, sometimes that's what the cell wants, but sometimes errors can cause problems there. And we know that there are diseases that result from errors of splicing, like Duchenne's muscular dystrophy, possibly Parkinson's, cystic fibrosis. So lots of things that can go wrong. All right, questions? Anything so far about transcription? All right, translation. Kind of sounds like transcription, but translating, again, like I said, into whole another language. All right, there are some steps that are similar. We name them the same as transcription. You'll recognize initiation, elongation, and termination. Those all are very different processes, though, in translation than what they were in transcription. We'll talk first a little bit about the tRNAs and their role, and there are a few modifications that we'll mention. But actually, first, we should talk a little bit about the genetic code. And this is not anything, certainly, that I would want you to memorize. I don't have hardly any of this memorized, but it's important to kind of understand and maybe even appreciate a little bit how we came up with this, so, and what it is. So these codons here, remember, these are codons or sets of 3 messenger RNA bases. Okay, this whole table tells us what amino acids are going to get made or added into a protein based on the messenger RNA codons. right? And how did we come up with this? Well, first we thought, well, maybe there's one amino acid for each base. And of course, that doesn't make sense because there's only 4 bases and we have like 20, 21 amino acids. Well, if we had, for two base pairs encoding for an amino acid, that wouldn't make sense either because there's actually only 16 combinations there. So we figured, well, we must have sets of 3. Because there's many more combinations, there is some degeneracy here. So, or sometimes we say wobble. So oftentimes, like for leucine here, that 3rd base of the codon, it doesn't matter what it is, it's going to code for leucine no matter what. So there's some wobble in that 3rd base. Of course, AUG is always the start codon that codes for methionine in eukaryotes or formal methionine in prokaryotes. And there's 3 different stop codons. And you don't have to have those memorized, but that's important. That's how termination of translation takes place. Okay, and deciphered by Marshall Nirenberg, 1961. It's been called the NIH's finest hour. He was in a race with a group at New York University to decipher the genetic code. Everybody, lots of other scientists at NIH dropped what they were doing and helped him with the sequencing experiments to do this. And he ended up winning the Nobel Prize for it. So, okay, all right, so how does the tRNA, of course, the tRNAs, their job is to bring the amino acids to the ribosome. And when the tRNA is loaded with that amino acid, we say that it's charged. Okay, so a charged TNA is carrying an amino acid. And just kind of briefly, how does that charging happen? Well, there's a particular set of enzymes involved with that, and they're called aminoacyl, if I can spell it, tRNA synthetase. Sorry, synthetase. This whole group, there's at least 20 of them, aminoacyl tRNA synthetases. What they do, it's actually 2 steps. They activate the amino acids and they attach amino acids to the tRNA. And of course, the amino acid, like I said, is added up, that acceptor stem that is called. All right, so again, this part is not in your book, but essentially this tRNA synthetase is, again, it's an enzyme that does a lot of different things. Let's use glycine as an amino acid because it's the simplest one. The first thing it does is attach an ATP to that amino acid. And I kind of wrote the steps out over here. It cleaves off 2 of those phosphates. Remember, when not just 1, but 2 are cleaved off, that makes it very energetically favorable. And that gives it the energy for the enzyme to attach it to the tRNA. Okay, so, go back to the other slide, yep. So essentially, what you end up with is the charged tRNA. Okay, so I just, it's a 2-step process. Basically, the amino acid gets activated with the ATP, and then it gets added onto the tRNA. What's the error rate of that enzyme? Well, it does have some self-editing functions. It makes a mistake actually quite a bit. 1 out of 40 times it makes a mistake and puts the wrong amino acid on tRNA. It's not too surprising if you look at the structures of, here's just a few of amino acids, the structures are really similar to each other, at least some of them. Of course, we've got some ringed amino acids, but it's not too surprising that it makes a lot of mistakes just because the structures are similar there. There is some self-editing to make the error rate more like 1 in 3,000, which again is still a lot more than the transcription or the replication, but that's okay because probably the next time the messenger RNA is translated, it will be done without errors. So, okay. All right, so initiation of translation. All right, so starting with your book's diagrams here. So remember, this is taking place at the ribosomes, and the messenger RNA, we're going to talk about how this has to line up just right on the ribosome. Our next graphic kind of looks at that a little bit more carefully, but it's actually lining up with the small subunit, okay? And the first tRNA that's carrying formal methionine, it always starts with formal methionine, is going to line up with the start codon in the P site. Okay, so usually we call that the peptide site because that's the site that of the small ribosomal subunit that holds the growing peptide. And the energy currency of translation is GTP. I'm not sure why that's used instead of ATP, but that's just the way it is. And once that first amino acid binds to the P site, actually the first one comes into the P site. After that, all the other amino acids come in to this A site. Again, that's, you can think of that as the acceptor site. There's a couple different ways you can remember that. Okay, and once that first tRNA comes in with the amino acid, that kind of signals the large subunit now to come in and form our initiation complex. Now we have a intact ribosome there, and new amino acids can start coming in. So basically that's initiation. I'll just write it here. There's also an E site, which is going to be right here, just stands for exit. So that's where the tRNAs go after they've unloaded their amino acid and they're ready to go out into the cytoplasm again. Okay. I brought this graphic in from another source because I like some details that it shows here about the initiation of translation. So, and some of this did not come through. Okay, so I'll have to redraw it again a little bit, that's okay. So the small subunits in this graphic is right here on top instead of the bottom. So that's a little confusing, but this shows the 16S strand of ribosomal RNA. Okay, and I like this because this shows us how it's really important that, this is again the messenger RNA, it's really important that that gets lined up just right with the ribosome, and actually what it's matching up with is this strand of 16S ribosomal RNA. And there's a sequence, so important that in bacteria, there's a sequence here that we give a special name to, the Shine-Dalgarno element, or Shine-Dalgarno sequence. Okay, so this is a sequence on the messenger RNA that matches to or lines up with the 16S ribosomal RNA. And it's important that that matches up exactly because this AUG has to line up exactly with the P site. Okay, if it's off by one base pair or another, then it's going to read this in, you know, the wrong reading frame. Okay, UGC, that doesn't correspond to anything, or it certainly doesn't correspond to methionine, formal methionine. So it has to line up just right there. Just erase that. Okay, So again, we give it a special name. Your book does use this term, Shine-Dalgarno element, in one place, but then it also calls it the ribosome binding site in another place. So just to be a little bit more clear about that. Yeah. So the question is the P site just for the formal methionine? That's where the formal methionine docks, but as we'll see during elongation, that's the site that's going to be holding the protein as it grows. So stay tuned there. Okay. All right, what else did I want to say about this? Oh, in eukaryotes, so eukaryotes don't have Shine-Dalgarno sequence, but that's where the 5' cap will line up just right, that guanine cap. And you can remember that it's the 5' site because this 3' end, let's see if I can draw this again. This is, here you got the R. This could be translating that messenger RNA in prokaryotic cells at the same time that it's being transcribed. So let's see. I'm trying to remember how I drew this exactly. I'm not going to be able to do it on the spot here. But so there's a strand of DNA here, and it's getting transcribed. This is really sloppy. Oh, and it's covered by a little label there. The point that I was trying to make here is that here's RNA polymerase. Okay, it can be transcribing DNA at the same time that the other end of the transcript is plugging into the ribosome. Okay, and that's always adding on to the 3' end. Right? So in prokaryotes, those two events can be going on at the same time. Okay, so let me just write that in. Transcription, translation can be simultaneous. Okay. Again, and that's only in prokaryotes. In eukaryotes, that transcription is off in the nucleus, so they have to be separate. Okay, so that's initiation. Really, so that just, initiation is just that first amino acid coming in, docking, the ribosome complex is formed, the large subunit joins the small subunit. And then elongation, again, we're back to your figures from your book here. Then a next, whatever corresponding tRNA with its amino acid comes in to the A site. Ribozymes in that ribosome act to form a peptide bond. So the peptide bond forms the protein, the new protein here is going to be associated at the A site just briefly, and then basically the messenger RNA kind of gets pulled through, and now it's said to be sitting again at that P site. Okay, and so on and so forth. The next, that first tRNA, after it unloads its amino acid, moves to the E site and then dissociates. Again, here's a new amino acid coming in and docking at the A site. The growing peptide gets moved over, and then it's sitting at the P site. Okay, this is all happening at a rate of about 15 amino acids per second. So it's kind of mind-blowing how fast this can actually happen. This YouTube video is kind of nice. If you start at about 2 minutes, the first 2 minutes talk about transcription, and then at about 2 minutes you have the transcript leaving the nucleus, so it is eukaryotic, but it snakes out through the nucleus, and then it's a cartoon basically, an animation, and it's showing the transcription and, sorry, translation and what it might look like. So again, the speed is impressive, kind of mind-boggling how fast this happens, how it all can match up and be correct. It's pretty amazing. All right, so just this graphic just reminds us, even though, sorry, my words got kind of smushed up there. You can get, especially in prokaryotes, we think this may happen in eukaryotes too, but probably not to the same extent. You can have lots of ribosomes all attached to the same messenger RNA transcript. Okay, so I talked about how the messenger RNA doesn't last very long, it degrades fairly quickly. If you want to get lots of protein out of that, lots of ribosomes can hang on to that same transcript and be making lots of protein there all at once. How does translation end? The book just kind of gives a little paragraph to that. I'm not sure if it even, I think there was an error actually in the figure that is there in the book. I'm trying to remember, I don't remember what the error is. But basically termination, okay, involves a stop codon, okay, on the messenger RNA. So UAA, if you check back in that genetic code, is one of the stop codons. So once that stop codon comes into the A site, there's no tRNAs that will match that. There's no amino acids that match that. So that causes a pause in the translation, and release factors bind to that messenger RNA. So these are proteins that come in and bind there and cause the disassociation. Basically the protein gets cut off and the ribosome dissociates and translation stops. Okay, so 3. Ribosome dissociates, all right? All right, so actually these, the release factors activate ribozymes, ribosomal RNA acting as an enzyme to cleave off that polypeptide chain. All right, so as with everything, we could go into a lot more detail, but this is kind of the main idea of how translation stops. And if you just remember that it involves release factors and a stop codon. Okay, transcription, remember, involved either row protein or some sequence in the DNA. And just, there's usually some kind of, or often some kind of modification that has to be done with that new protein. Oftentimes, it's not just gonna right away start working in the cell. For whatever reason, in prokaryotes, usually that formylmethionine is cut off. Perhaps part of the reasoning there is because that will, it's kind of a red flag that this is a prokaryotic protein. So that's probably why it's cleaved, because it would be recognized by our immune system really easily as something as foreign. So the formylmethionine is removed. In eukaryotes, the methionine would be removed as well. So not sure why that happens. If it's an enzyme, if the protein is an enzyme, then there may be some kind of cofactor or coenzyme that's necessary for it to be functional. There may be some different folding that needs to happen, or quaternary structures when two polypeptides are kind of formed together and bonded together. And there may be some kind of snipping or cutting that needs to take place, because the cell may not want that protein to be active inside of the cell, especially if it's a toxin. Okay, you don't want it to function inside the bacterial cell. You want it to start functioning after it's released, after it's secreted. So there's all sorts of maybe things that need to be done at just the right time to make that protein functional. Okay, and we always like to compare things. So let's see, I think I said some of this already, but if we compare transcription in eukaryotes and prokaryotes, of course, in eukaryotes, we get one gene for one messenger RNA. In prokaryotes, like I said, it's polycistronic. We had the 5' guanine cap on the messenger RNA binding to that small subunit of the ribosomes to initiate. So that's going to be the 40S in eukaryotes, is a small subunit of the ribosomes. Remember, we had a specific sequence that binds there, the Shine-Dalgarno sequence of the messenger RNA binds to the ribosome, small subunit of the ribosome. Of course, in eukaryotes, transcription takes place in the nucleus, translation at the ribosome. Okay, transcription and translation are both in the cytoplasm. Of course, the ribosomes are in the cytoplasm. Okay, and formal methionine is the first amino acid instead of methionine. Okay. All right, so I think I will just leave you with today. Oh, before I do that, a couple other things. This is a really nice figure in your book that they may have left off in the most recent edition. I think that's why I put there 5th edition. But it kind of helps you understand, okay, written in the DNA strand, right? There's a promoter region that's necessary for transcription. Remember, that's where the RNA polymerase binds. The new messenger RNA transcript has that start codon. Again, we called this the Shine-Dalgarno sequence. You could also call it the ribosome binding site. And basically, that gets transcribed, the messenger RNA at the ribosome, into its different proteins. It's a good table that your book has to kind of sum up these 3 processes: replication, transcription, and translation. A really good way to study is to cover part of it up and see if you remember what the difference is between them. We didn't really talk about this down here, so you can kind of forget about that and don't memorize those SOT codons, but it's a great way to study. So we'll pick up next time with regulation and hopefully start the viruses on Tuesday, so I'll get that information posted. Have a good weekend.
Can you go back a couple slides?
Yes. That one. Is that one? Okay.